A-Level Biology 9700, Paper 4, Variant 2, October, November 2024, question number 7. Paper chromatography is a technique that can be used to separate and identify different chloroplast pigments. Describe how the results of paper chromatography can be used to identify chloroplast pigments. And students, they have told us that is the paper chromatography is a technique that can be used to separate pigments based on their different solubilities in the solvent and their affinity for the paper. So, first of all, how the results of paper chromatography? It means after paper chromatography, the results which we obtain, how these results can be used to identify chloroplast pigments. First of all, uh, when the paper chromatography occur, after paper chromatography, the solvent moves up in the chromatography paper. So the different pigments which are present in the chloroplast extract will travel at different rates. So the chloroplast pigments will travel on paper by different rates. So, and secondly, we can, and this will result the separation of the pigments into different distinct color bands or spots on the paper. So this traveling will lead to the different color bands. Or spots. Different color bands or spot will be appeared on paper chromatography, with the help of which we can identify the different pigments which are present in the chloroplast. So according to this, the different colors which will be shown by these different pigments, the carotenoids, they will appear as orange or yellow bands. So it will show a distinct color orange or yellow. Second, xanthophyll. So the xanthophyll, it will appear as yellow. It will appear as yellow or brownish yellow. And the third, chlorophyll A. So chlorophyll A will appear as blue-green band. And the fourth one, chlorophyll B, it will appear as yellow-green band. What is happening during the traveling, during the uh, travel of these different pigments with the solvent? So they will show their distinct colors, as carotenoids will show the orange or yellow bands, xantho will show the yellow or brown, yellowish brown, and the third chlorophyll A will show the blue green band, and chlorophyll B will show the yellow green band. So in this way, the different colors can show us the results or the different pigments which are present. Second method, would like to write over here to calculate the retention factor. Retention factor is RF value. So RF value is the retention factor. So the retention factor can be calculated for each separate pigment band. So it is the ratio of the distance traveled by the pigment to the distance traveled by the solvent front. So I would like to write its formula. Maybe it is also written in your marking scheme, but this is the authentic method, that is distance moved by pigment divided by distance moved by solvent. So for this purpose, you will have to measure the distance which will be shown over the paper chromatography paper, and then the distance which will be moved by the solvent. You have to calculate these values, and by this formula, you can find out the difference between the different pigments. So the difference between the different pigments will show the movement of different types of pigments over here. So, as the each pigment has a relatively consistent RF value when the experiment is under the same experimental conditions, such as the same solvent, we are using the same solvent, we are using the same type of paper, and under the same temperature. So, in this way, these two methods are helpful to find out the uh, to find out the or to identify the chloroplast pigments during the paper chromatography. Thirdly, if we want to identify the pigments, the RF values obtained from the chromatography of the known chloroplast extract are compared with the RF values of the known standard pigments run under the same conditions. So we can write that by comparing the obtained RF values, during the you could say with the values of non-standard pigments, of non-standard pigments. So in this way, and the the color is also consistent. So first method, the color is very consistent. So with the help of the color, we can find out the or we can identify the different chloroplast pigments. Secondly, by calculating their RF values, and third, by comparing their RF values with the other standard with the values of the other standard pigments. So in this way, we can find out, we can the answer of this question. So I have also written this paper chromatography allow for the separation of chloroplast pigments by observing the color of the separated band, as I have written the different colored bands over here, and calculating and comparing their RF values to non-standard. The different chloroplast pigments present in the original extract can be identified. So these are very simple methods with the help of which we can identify the different chloroplast pigments. Now I am moving towards the next question. And next question is carotene and xanthophyll are chloroplast pigments. So here question is describe the role played by these pigments in photosynthesis. Students, photosynthesis. Photosynthesis is a process in which the green plants means the plants which have chlorophyll inside. So the chloroplast or the different chloroplast pigments are used for the process of photosynthesis. The process of photosynthesis cannot take place in the absence of chlorophyll, or where the chlorophyll is not present in the plants. So the process will not take place because these pigments or chloroplast pigments are used for different purposes during the process of uh, photosynthesis. So first of all, their main function which we study from very junior classes, that is the chlorophyll is a grease pigment which is used or which captures the sunlight for the process of photosynthesis because it absorbs. So the different, it absorbs the light energy in the regions of the spectrum that chlorophyll does not absorb well. So I would like to say that xanthophyll, the role of carotene and xanthophyll is that they absorb light energy, light absorbance. So the first important function is light absorption or light absorbance. They absorb the light energy in the regions of spectrum that chlorophyll does not absorb well. So I would like to say that these two pigments broadens the range of the light wavelengths that can be used for photosynthesis. So increasing the overall efficiency of light harvesting. So light absorbing, or you can say the efficiency, increasing efficiency of light harvesting. Harvesting or capturing the light. So the absorbed energy is then transferred to chlorophyll, a molecule in the reaction center. So after absorbing this, uh, the energy which is absorbed by these pigments, so is transferred to chlorophyll A. In the reaction center. Reaction center. And in the reaction center, it is used to drive the light dependent reactions. Used to light dependent reactions. Such as photolysis. So first of all, the first function is the light absorbance or absorption of light, which increases the efficiency of light harvesting. So the chlorophyll A, this energy is transferred to the reaction center, and then that energy in the reaction center, that energy is used for the light dependent reactions such as photolysis. Photolysis, during which the water is broken down into hydrogen and oxygen. So this is, it takes place in the light dependent stage in which light is compulsory. And then second important function is the photo protection. Photo protection. Photo protection means, what does it mean? Photo protection. So carotenoids, including carotene and xanthophyll, they play a vital role in protecting the photosynthetic apparatus from damage caused by excess light energy. Sometimes there is excess light energy. So protects the damage, protects the damage from excess light energy. You know, most of the energy is used for the process which takes place in the light dependent state. So the excess energy can damage the photosynthetic system or photosynthetic apparatus. So they protect the plants from the damage which can occur by excess light energy. So when light energy is absorbed by chlorophyll exceeds what can be used for, it can lead to the formation of harmful reactive oxygen species. So excess energy can lead to excess energy can leads to the formation of harmful harmful reactive oxygen. So these pigments uh, prevent the damage which can cause by these reactive oxygen species. So in this way, they protect the plants, they protect the photosynthetic parts of the plants. And third, I would like to say, the third function I would like to say that is they can directly scavenge and neutralize harmful ROC such as silent oxygen. So they also play a structural role. So carotenoids are also structural components of the photosystem. Carotenes and xanthophyll are also structural components. Components of photosystem 1 and 2. So in this way, I could like to say they help in stabilizing the pigment protein complexes within the thylakoid membranes, ensuring the efficient organization and function of the photosynthetic machinery. That is the three functions I have written. The first one, the light absorption, they help in the light absorption. So the light absorption, absorbance increasing the efficiency of light harvesting. So when the chlorophyll A, then this energy which is absorbed is transferred, it is used into the reaction center, and then in the reaction center it is used for the light dependent stage. That is you so the most important process takes place in the light dependent stage, that is the photolysis. Secondly, the photoprotection, that is during when there is excess light energy that can be harmful for the photosystems, as for example the excess energy can lead to the formation of a harmful photoreactive oxygen species. So carotenes or carotenoids, they protect the photosynthetic apparatus from the damage which can cause by this uh, reactive oxygen. And the third one is the carotenes, carotenoids, that is carotene and xanthophyll, are also the structural components of photosystem 1 and 2. So in this way, I like carotene and xanthophyll are essential chloroplast pigments that not only contribute to light harvesting and also play a crucial role in protecting the photosynthetic apparatus from photo damage, that is the excess energy. So ensuring the survival and optimal functioning of photosynthetic organism, these two pigments play a very important role. Now I'm moving towards the questions. The light-dependent stage of photosynthesis produces ATP and reduced NADP, which are used in the light-independent stage. So describe the light-independent stage of the photosynthesis. Light-dependent stage and independent stage. These are the two stages which takes place during the process of photosynthesis. The light dependent stage, in which the light is absorbed by the chlorophyll, then that light energy is changed into chemical energy, then it is sent to the reaction center. Uh, and in the reaction center, the photolysis takes place, and during the photolysis, the water is broken down into hydrogen and oxygen. And thirdly, the second stage is started the um, in which the formation of carbohydrate takes place. And during the light independent stage, light energy is not required because there is no need for light now, because already light has been absorbed during the stage of light dependent stage. No, here there is no dependence on light. So light independent stage, during the light independent stage, the chemical energy is stored in ATP and ADPH from the light dependent stage to fix carbon dioxide and produce organic molecule. Organic molecules, the carbohydrates, which are formed during the process of photosynthesis, so that the plants can use for energy and growth. So that this process occurs in a cyclical pathway known as the Kelvin cycle. The whole process or the whole light independent stage, the breakdown of the steps of Kelvin cycle, which I have written over here. The first is the carbon fixation. So what happens during carbon fixation? Carbon fixation. So carbon fixation is very first step which takes place during the uh, carbon fixation stage. The carbon dioxide from the atmosphere, atmosphere enters the stomata. Carbon dioxide enters the stomata and combines with this carbon dioxide. It will combine with 5 carbon molecule called as ribulose 1,5 biphosphate. 1,5 biphosphate. So during the, which is also called as RUBP. So this is the, so this reaction is catalyzed by the enzyme. Which enzyme is used over here? That is RUBIS, Robisco. So ribulose by 1,5 biphosphate carboxylase enzyme is used for this process. So resulting 6 carbon compound, formation of 6 carbon compound. Carbon dioxide will react with ribulose 1,5 biphosphate, and there will be the formation of 6 carbon compound. And the enzyme which will act during this is the Robiscose, or you can say Robisco 1,5 biphosphate carboxylase enzyme. So after the this formation, 6 carbon compound, it is very unstable compound. It is very unstable compound, so it will split up. It will split up into 3 carbon compounds, which is 3 phosphate glycerate. 3 phosphate glycerate. So there will be 2 molecules of 3 phosphate glycerol. So after this 3 phosphate glycerate, here there will be the, due to ATP. After next stage, in the next stage, what will happen? By the formation of glycerate 3 phosphate, then what will happen? There will be the 2 molecules of this 3 phosphate glycerate. Then the reduction will take place. ATP and reduced NADP form the light dependent reactions are used to convert 3 phosphate glycerate into another 3 carbon compound called glyceraldehyde 3 phosphate. So ATP plus NADP. So they will help in the formation of 3 glyceraldehyde, glyceraldehyde 3 phosphate. because first of all, it is phosphorylated by ATP in the first step. So in the first step, ATP phosphorylated by the ATP. So and in the second step, uh, the 1,3 phosphoglycerate glycerate is then reduced by NADPH, losing a phosphate group become G3 glyceraldehyde 3 phosphate. So it will be can be written in this way. First of all, what will happen? Phosphorylation takes place, and this by the ATP. So there will be formation of 1,3 biphosphoglycerate. Then 1,3 biphosphoglycerate is reduced by NADPH, then losing a phosphate group to become glyceraldehyde 3 phosphate. So after this formation, the regeneration of the ribulose for the Calvin cycle to continue. So then there will be the regeneration of RUBP. So in this way, this whole process takes place in the form of a cycle. So during this, I would like to explain, first of all, the carbon dioxide from the air, it will enters through the stomata, and then it will ribulose by 1,5 biphosphate, and after reacting with ribulose 1,5 biphosphate, there will be the formation of 6 carbon compound, and then which is very unstable compound. So it will break down into 2 molecules of 3-phosphate glycerate. Then ATP and NADP, which are from the light dependent state. So this phosphorylation by ATP will take place, then it will be changed into 1,3 it will be changed into 1,3 biphosphoglycerate, and then by the NADP, it will be changed into glyceraldehyde 3 phosphate compound. Then regeneration of the ribulose biphosphate will take place, because this is a Calvin cycle. Cycle means it is repeated again and again. So Calvin cycle to continue, ribulose biphosphate must be regenerated. So some of the glyceraldehyde 3-phosphate molecules are used in series of reactions that with the help of ATP arrange them back into the ribulose phosphate. So in this way, it allows the cycle to begin again with the fixation of more carbon dioxide. Products of the light independent stage are glucose and other carbohydrates. Products, I can write over here, products of light independent stage are carbohydrates, glucose, and other carbohydrates. So some of the glycerol 3-phosphate molecules produced are used to simply in abbreviation G3P to synthesize glucose, which can then be converted into other carbohydrates like starch for storage and sucrose for transportation, and other organic compounds which are the intermediate molecules of the Calvin cycle can also be used to synthesize other essential organic molecules such as amino acids and lipids. Now this process takes place in the form of a cycle. This is called as the Calvin cycle, which is repeated again and again, because during this process, the formation of carbohydrates or the other glucose, and especially the glucose and the other carbohydrates will take place. And generally, these carbohydrates are converted into lipids and amino acids as well. So it was all about question number 7, and thanks for watching. Please like, share, and subscribe my channel. If you have any query, you can message me. Thanks for watching.